Alexa Knoll,
I'm Abby.
and this is our research presentation for anatomy and physiology. So first we will get started with a broad question.
So our broad question was, how do prion diseases and fatal neurodegenerative disorders like mad cow disease contribute to synaptic degeneration?
And here we have our targeted research question for our experiment, which was how does P38 inhibit the pathway for PRPSC to produce neurotoxic signals resulting in dendritic spine degeneration?
We're going to move on to our background information. And prion diseases are basically fatal neurodegenerative diseases such as bovine spongiform encephalopathy, which is mad cow disease. And prion diseases basically cause synaptic degeneration and ultimately the loss of that synapse. And synapses are the structures that connect nerve cells, and they play a vital role in learning, memory, and neurological diseases.
Okay, and now for molecular mechanisms. PRPC is a naked protein molecule which is needed for neurological signaling. However, there is a mutated, altered, infectious form of this protein called PRPSC. When prions propagate, the PRPSC molecule force their beta sheet reach conformations on the PRPC molecule. This will initiate a synaptotoxic signaling cascade and begin the activation of the NMDA receptors. In this experiment, P38, which is a mitogen-activated protein kinase, is shown to be an inhibitor of the PRPSC signaling cascade. Why is this important? This research is important because it affects many people and animals with the prion disease everywhere. Researchers previously discovered that the source of rapid retraction of the dendritic spines is purified PRPSC. The exact cause of the retraction of the dendritic spines is still unknown and needs to be further researched. However, with the data from this study, pharmaceuticals can be distributed to patients everywhere with dendritic spine degeneration and loss. This research gives new insights to Alzheimer's disease and mad cow disease. Prion diseases have a major impact on the ability to learn and memorize information.
Okay, so next I'll talk about the materials and methods. So we experimented on wild mice. So basically, we measured the P38 mark phosphorylation in the dendritic spines, and we also used low-density hippocampal neurons, and we used those to measure the dendritic spines to compare the number of them after the experiment. We also treated the neurons with purified PRPSC and stained them so that we could visualize the dendritic spines and axons. And for each experiment, we used 15 to 24 neurons from 3 to 4 individual experiments that were imaged and quantitated.
Okay, so next we will discuss our results. So the PRPSC causes functional changes in synaptic transmission. Normal dendritic spine retraction occurs with the expression of the PRPC molecule. The expression of the PRPSC molecule causes a reduction in the miniature excitatory postsynaptic current, or MEPSC, frequency and amplitude.
We also found that the PRPSC did not significantly affect the frequency or amplitude of the miniature inhibitory postsynaptic currents. Also, PRPSC is very selective in its effects. It targets primarily excitatory, not inhibitory synapses. We also used a culture system slightly different from the one used to monitor the dendritic spine retraction, and that also caused a reduction in the MEPSC amplitude and frequency.
Okay. PRPSC synaptotoxicity requires a glutamate receptor activation and is accompanied by the calcium influx. Both the competitive AMPA receptor and uncompetitive NMDA channel blocker prevented the PRPSC molecules from binding, which caused an induced retraction of the dendritic spines. The number of dendritic spines in the infected and non-infected mice were relatively the same.
From the results obtained from the inhibition of the P38 MAPK, we used a genetic method to suppress signaling. Resulting from that, there is a double mutation in the activation loop of the kinase that prevents phosphorylation of upstream kinases. And this has a dominant negative effect on the activity co-expressed by the P38, which significantly attenuates signaling. This method of reducing P38 signaling avoids the embryonic lethal phenotype that results from the complete inactivation of the P38 MAPK gene.
And now for our discussion. Critique of the study. In this study, we could have better explained the neuron interactions and could include more details of the PRPC and PRPSC molecules.
I think we could also make the research paper a little bit more specific. It was very specific in the results, but not as specific everywhere else, so I feel like we could do a little bit better with that.
And next, for our future experiment, our next step is to conduct an experiment to see if the P38 MAPK can affect the actin in the cytoskeleton, which could be another potential pathway for the PRPSC, which could cause dendritic spine morphology due to the activation of the kinase.
This research can be applied in a number of places, such as a lot of neurodegenerative diseases such as Alzheimer's, dementia, and mad cow disease. This research helps for the betterment of the understanding of what goes on in those diseases and how proteins might be able to stop or even reverse them.
As previously mentioned, mad cow disease is one application of the research study that we conducted. Mad cow disease is a prion disease. It is an infection in the brain of the cattle. The brain of the cattle, after it's had this infection, makes it have a sponge-like appearance. And this infection leads to deterioration of the cow and later death. It can be dangerous when humans ingest this form of infection from cattle meat. And the human version of mad cow disease is called Creutzfeldt-Jacobs disease. And this is the source for our article. And thank you.